push. There we go. I'd like to thank everybody for coming to the Cancer Center Symposium. I'm Matthew Reith. I'm one of the medical oncologists here. I'd like to introduce Dr. Cameron Rathmell. She's the Cornelius Abernathy Craig Professor of Medicine and the Director of the Division of Hematology and Oncology at Vanderbilt University in the Department of Medicine. She's a physician scientist studying the molecular biology of renal cell carcinomas and rare tumors of the kidneys. She earned her undergraduate degree in chemistry and biology at the University of Northern Iowa and her MD and PhD in biophysics at Stanford University. She trained in internal medicine at the University of Chicago and the University of Pennsylvania and fellowship in medical oncology at Penn as well. She joined the Department of Medicine and Genetics at the University of North Carolina in 2003 and was a founding member of the Department of Urology before coming to Vanderbilt in 2015. She helped lead the Cancer Genome Atlas working groups for the study of renal cell carcinoma and is co-chair of the Kidney Chromophobe Analysis Working Group and the current Pan-Kidney Working Group. Clinically, she conducts diagnostic and therapeutic clinical trials in a clinic devoted to renal cell carcinoma at Vanderbilt University. And we appreciate her taking the time to come and talk to us about translational studies in renal cell carcinoma.
Yep. Thanks. Can you hear me okay? In the back? Okay. So thank you for the introduction. I'm really delighted to be here. It's beautiful weather to come and see here. I love your campus, and it's really fun to be invited by one of our former fellows and seeing Matt doing a really great job here getting started. So I've had a great day, and happy to talk to you about some of the research that we're doing in the laboratory. So as Matt said, we study kidney cancer, or renal cell carcinoma. I say the renal cell carcinomas, a few disclosures, because this is a complex set of diseases, although most of what I'll talk about today is the major subtype, clear cell type renal cell carcinoma. So a few of the slides are in here a little different from my traditional science talk because the last talk that I gave was at the AACR Molecular Biology and Clinical Oncology Workshop, where you're supposed to filter in a little bit of why you do what you do. And I made this slide for that, which was why I study kidney cancer. There's more money in breast cancer. There's more access to tissue in heme malignancies. And so why kidney cancer? And so I fell into kidney cancer, and I'm here because there is enough to keep me here forever. And I think what you might see in this talk is that we've worked pretty diversely. We got started in VHL and HIF biology, studying hypoxia and metabolism. And I'm not going to talk about that today, although we have active projects in metabolism and pseudohypoxic state. The new epigenetic and chromatin modifiers have opened up a whole new world of possibilities of things that we can investigate, and I'll talk about that. And then tumor immunology, everybody does tumor immunology now, and so we do as well. And as I mentioned, it's a disease of multiple cancers, and so with the very rare subtypes, it's a splitter's paradise. So when we say renal cell carcinoma, usually we mean clear cell type renal cell carcinoma, which is, I know I saw a pointer. That's all right. Which is so named for its clear cell appearance, the clear cytoplasm. But other common variants are papillary type renal cell carcinoma. That makes up about 20%, so not uncommon to see this subtype. And chromophobe, which makes up about 5%. But there's a lot more. In fact, there are over 25 WHO-designated subtypes of renal cell carcinomas or carcinomas that arise in the kidney. So we've had a lot of fun splitting out those subtypes into ways that you can subtype clear cell into different prognostically relevant and molecularly relevant subgroups. The papillary type is really interesting. There are at least 4 very clearly defined molecularly different subtypes that lump into the papillary type, and chromophobe also has some of its own, plus some very rare variants. But again, for today, I'll put my splitter hat aside, and we're going to talk about clear cell. I forgot this slide also to say, though, that with all this splitting, the truth is that we largely treat these patients all the same. The VEGFR TKIs really have been tested and can have activity. That's the best that the data is in the non-clear cell subtype, and so we use them. And then the use of immunotherapy seems to cross an awful lot of boundaries and is really going to be a game changer, even more so in kidney cancer with data that just came out on Saturday with ipi-nivo combination immunotherapy improving survival over sunitinib, which is our standard anti-angiogenic. So Immunotherapy is going to take the lead now, although I'm sure the anti-angiogenics will still stay there. As I mentioned, actually I'm not going to spend time on this. This is the basic differences in renal cell carcinoma. Again, we're going to focus on the clear cell type in the light blue. So almost all of these tumors have a mutation in the von Hippel-Lindau gene, so they're VHL deficient. That leads to upregulation of HIF, the hypoxia-inducible factor, and a host of phenotype that comes after that, because this is a transcription factor that turns on genes involved in metabolism, cell migration, cell survival, Wnt signaling, et cetera. So, and even far-reaching things like erythropoietin production, so that you increase red blood cell mass. So there's a lot that can be done there, but we know that that's not enough to make a cancer. It doesn't work in mice, and in people, they consistently have more than just a VHL mutation. They tend to have these epigenetic modifier mutations too. So this is the Cancer Genome Atlas overview of the clear cell type renal cell carcinoma Sept. And what you can see here, let me try this just one more time. Let's see if this will. Does anybody know where the pointer? Somebody showed me a pointer, and then it's a little bit hard to gesture from here, although I could just do this. No, that's okay. Yeah, I'm not sure that's pointer. Yeah. All right, I'll do this. Does that show up there? Is there a? Yes. Okay, that will work. So this is a heat map of the clear cell renal cell carcinomas. Mutations shown here in light blue. So VHL is on the top. VHL identifying mutations in about 60%. There are some technical reasons why we think that there are more mutations than that as well. VHL is located on chromosome 3p, and you can see 3p deletion almost universal. That's the top blue line here in the SNP analysis. So the next most commonly mutated genes are all these epigenetic modifiers. So SETD2, BAP1, PBRM1, KDM5C. Most of those are also on 3p. So again, mutation with obligate loss of heterozygosity is sort of this defining feature. And with all of these things located on 3p, what you can appreciate is that this is a unique setup for this particular tumor type. And it's also why it's been more challenging to see this tumor probably in mouse or other animal models, because these sections of chromosome 3 are dispersed across the mouse genome. So within that small segment, VHL, BAP1, SETD2, PBRM1 are all co-localized, and all in a region that's deleted. So here we see the deletions from the TCGA group shown in blue. There's not a particular breakpoint, but they always lose at least up to the section where the rest of the chromosome contains all these genes. So we knew they were important from that perspective. The other high-throughput sequencing experiment that really nailed that VHL is the truncal mutation and these others are required for the next events was multi-region sequencing that started in about 2012 in renal cell carcinoma, where renal cell carcinoma was identified early on as this very heterogeneous tumor. So if you sequence across multiple regions, first groups in the UK and then other groups, and ours also looked at as well, identified that what you find is that you'll consistently find the same VHL mutation, and you'll consistently find the same 3p breakpoint. So they're clonal at that level. So all of these little blue sticks all have a VHL mutation. They might have another mutation as well. And that's what's common to all of these tumors. So that really defines this as truncal. But then all the tumors have additional and discrete mutations, and that helps to define what those second hits are. And so we have numerous second hits that are repeated and repeated within branches. So this allows us to detail the primary and secondary mutational events in renal cell evolution. And in particular, we study SETD2 because it really shouted out in these heterogeneity studies. So here's an example of a tumor. They sequenced from multiple regions of the primary as well as the metastasis. And you can see VHL mutation at the trunk, like we always know. And then as they branch out, you can see this branch has a SETD2 frameshift mutation. This branch has a SETD2 missense mutation. This little branch has a SETD2 splice site mutation. So to me, this is saying that this tumor wants to have a SETD2 mutation, that it's important for this tumor to have lost that second copy of SETD2 in its function. And so that's what really led us to choose that one of the other chromatin modifiers that are mutated in kidney cancer, although there's good rationale to study BAP1 or PBRM1 as well. But this is what got us the most excited. So SETD2 is one of many chromatin modifiers. So this is a simplified schematic of chromatin DNA wrapped around a nucleosome and the histone modifications that are dictating where different reader molecules come and sit and interpret what the intention is of that region of the chromosome or chromatin landscape. And so SETD2 is a non-redundant histone methyltransferase. It puts down the third methyl, the trimethyl of lysine 36 on histone H3. So H3K36 trimethylation. Mono and dimethylation are laid down by other enzymes. This only does trimethylation, and there is nothing else that does trimethylation. So that was actually a really convenient thing for us to be able to look at. So I probably should set this up before I move on to the slide, because you look at it and go, what are they doing here? So here we are. We're a VHL HIF biology, molecular biology laboratory with this new finding in 2010, 2011, that there are all these chromatin modifier mutations. And we really know we have to get into that. And so conveniently, the laboratory next door was a chromatin lab. It was a pediatric oncologist who studies Wilms tumor and also Ewing sarcoma. And this is Ian Davis at UNC Chapel Hill, and was also getting started on doing a genomic landscape approach to look at more open and closed chromatin. And so with him, along with Jason Lieb from the Department of Genetics, we decided to take a really simple, turned out it was not simple, but a simple-minded approach, saying we don't know what these things are doing. We've lost histone H3 trimethylation. It must be doing something. Must be doing something to chromatin. How do we look at chromatin? So we took human tumors, clear cell type renal tumors, and we did a process called FAIR-seq. So FAIR-seq is a little like ATAC-seq, for those of you who know that. ATAC-seq hadn't been discovered yet or invented yet, but FAIR-seq uses a physical separation based on basically a phenol chloroform extraction of sonicated chromatin. So you're separating out chromatin condensed and chromatin open. And then you do short read sequencing and see what's open. And so we thought, this will be simple, and this will tell us something. It was hard, and it was not as clearly informative as we thought it would be, but it clearly told us that tumors are different than normal. And so that's what we see here. So this is the FAIR signal across these tumors, and across over on the right, you can see some matched normals. There were a lot more tumors than we had matched normal for. And each of the yellow marks is a 500 base pair interval that was identified as open by our short read sequencing. And so you can see very clearly that tumor is different than normal, that there's a lot more open than there is in normal. And when we go diving into what are those regions, one subset, that cluster 1 at the top, was a nice validation. It was what we found. There was a lot of promoter regions or transcription factor binding sites, which is what FAIR-seq has been utilized to discover in cell lines. So it actually worked in whole tumor, which we really weren't sure it would do. And it found things like the transcription factor binding site for HIF. So we know that HIF is up. We know that those sites should be open. They should be available, and they were there. So that's great. And then we found a whole lot of other stuff that was open. And we really had a lot of trouble classifying exactly what all those open things were, aside from those that were important for the transcription factor motif. So we turned back to SETD2. So we know we have an interest in SETD2, and we know that it's dropping H3K36 trimethylation. It binds to RNA polymerase II and travels with the transcription complex so that it methylates on actively transcribed genes. The thought is actually that that methyl group is inhibitory of transcription, but of course, it's in actively transcribed genes because it's coming along with RNA Pol II. The idea from yeast is that it's there to protect cryptic initiation. So it's there to say, this part's already done. Don't come in and do it again, okay? Or at least that's sort of the simple thought of what this mark is doing, why it's a repressive mark on actively transcribed genes. So it does a bunch of other stuff as well. And that was also intriguing to us. It also, this mark is bound by DNMT3A and 3B, so it's involved in DNA methylation, it's involved in DNA repair, and can bind to DNA repair machinery, and it can also, in yeast, be involved in transcript elongation and also processing. We were also interested in whether or not we could see differences in, if you lose one mark, there can be compensatory marks as well, and whether or not the altered histone mark placement would alter DNA methylation, transcript processing, or chromatin accessibility. So it gave us some work to do. So we classified these tumors two different ways. We did do sequencing for SETD2 mutations, but we also stained them for this H3K36 trimethylation mark. Conveniently, the antibodies there are really very good and very specific for this very unique little mark. And so what you can see here is an example, my label's gotten lost. In the top is from a patient who has a SETD2 mutant tumor. And so the histology there doesn't show any brown mark. That's absence of H3K36 trimethylation. You can see individually little spots, and those are non-tumor cells that are present in the tumor. As compared to the uninvolved kidney of that patient, then there's lots of brown marks because all those tumors are transcribing something, and so they have H3K36 trimethylation. And most tumors that have a wild-type SETD2 will have plenty of methylation. And you can see the correlation there between mutant SETD2, shown in red, having the lowest levels of H3K36 trimethylation. So more of the tumors were screened by methylation than by sequencing because of available tissue. But we did confirm that. And this is why another reason why we picked SETD2 to work with, because we could very accurately put tumors into two different bins. So now we go back to our FAIR-seq data and look at it in relation to the SETD2 mutation or methylation status. So here we have a set of tumors, red indicating that they're SETD2 mutant, gray, SETD2 wild type, white didn't get to be sequenced, and looked at the FAIR-seq data. And here it became more clear that some of this open chromatin that we were seeing before is being driven by the SETD2 mutation. So we have, again, a large number of 500 base pair genomic intervals that are scoring positively for being open in the SETD2 mutant tumors as compared to the others. So they didn't have a great signature. They weren't promoter or enhancer regions that were annotated previously. But a lot of them were regions over exon starts and were close to splice site junctions. And we also noticed that we were getting a lot of, in our transcript analysis, we were getting a lot of introns reading in our transcripts. We were doing ribosome depleted transcript analysis, not poly-A selected. And so we developed a score for retention of intron in the transcript pool. And it turned out that in those tumors that were H3K36 deficient or SETD2 mutant, that we had a striking increase in the number of retained introns in the transcript pool. Other errors of splicing were also significant. So this is a graphic showing sort of significance testing. Anything above 2 is a greater less than p.05. For alternative splicing, the intron retention shown in blue, and alternative transcript start and stop sites. And so you can see we were seeing a lot of errors in this pool. Interestingly, though, it didn't make up the majority of the transcripts. Obviously, these cells were still alive and they were still kind of kicking. So for any given transcript, this represented less than 1% of the total transcript pool, but significantly much above normal. So when we integrate this analysis, focusing on the exon start sites, we thought we could at least assume why it is that we're getting some slippage in the accuracy of splicing here. And what we think is happening is that we're removing a nucleosome that should be well positioned at the start of an exon. So going to walk you through this data a little bit slowly. So basically, if you just look at one line at a time, it's very helpful. So first, let's look at the gray line. So the gray line is a chip for H3K36 trimethylation. So H3K36 trimethylation is on actively transcribed genes. It's usually preferentially at the start of exons. And you can see that, that here we're lining up all of the exons that we had recorded in missplicing. So this is thousands of exons that are all compiled vertically. And looking at the H3K36 trimethylation signal, and you can see a big peak. That's the gray line right over the start. So that's normal. We expect to see that because there should be a nucleosome there, should have a histone H3. It should be trimethylated if it's a transcribed gene. Compare that over to the random genic position. You don't see that spike because here the nucleosomes are going to be more dispersed. And at a random internal exon start, we do see the spike. So now if we look at the black line, this is in a tumor that was normal for H3K36 trimethylation, SETD2 wild type. And looking at our FAIR-seq signal, so this is the open versus closed chromatin. And there's normally a little dip right over the exon start site because there should be a nucleosome there, so it should be bound. So this should be not as open, and that was also true for the random internals. But when we have our SETD2 mutant H3K36 trimethylation deficient, then suddenly we see a spike there. So the nucleosome is preferentially off of the start, and so what we think is happening is that you have exposed a region that normally is probably managed with some great care in the process of transcription, and displaces the nucleosome. We've tried to really prove that with MNA-seq and see really where are the nucleosomes, because then you can get a really discrete picture. We can see for sure that there's some wobble, and I don't have that data here, but we think that on selected exons that there's sort of a shifting of the nucleosome, and that's why you can get some more splicing. So, sorry, I just told you the model. So the model is that, normally with H3K36 trimethylation, you get proper splicing, and then when it's gone, either because it doesn't have a signal for the spliceosome machinery to find the proper splicing spots, or because the signals are different and are indicating maybe that you shouldn't be splicing there. And either way, you get a large number of splicing defects. So, but is that enough to cause cancer? Yeah, that paper was in Genome Research. It would have been in Nature Medicine if we had proved that caused cancer. It's a thing that happens, but we're still not entirely convinced that it is the root cause of kidney cancer from losing SETD2. We're still not sure what that is, but we have a little bit better idea now from an experiment that was a little bit serendipitous. So we were working with some panel of cell lines where we knocked out SETD2. We knocked it out with the TALEN system, which is like CRISPR, but not as cool. And we had a collaborator friend who knocked it out in a mouse model. And so she had an inducible mouse model. So she could knock out universally very quickly and look at acute loss. We were knocking out and then having to select and then flow sort. And so we knew our cells weren't very happy when they were first knocked out, but they all recovered. And several weeks later, we'd have a nice clone, right? You could make a pool, but we were picking clones. And so we had these clones that our cells were fine. In fact, in data I'm not going to show, our cells are quite fine. So knocking out SETD2 gives them a proliferative advantage. We don't really know why chromatin modification would do that. They also migrate faster. So they're really a much more aggressive cell line after the fact, after we survive. She was seeing something quite different. She was saying when she knocked out SETD2, her cells just die. And so we started really looking at these cells. And unfortunately, my movie's not going to go. But what we were seeing was with acute loss, a lot of the cells were really having trouble dividing. And so what the movie would show, if you go through, so the single panel pictures the movie, is a cell that goes to round up, like it's going to divide, and it waits too long. We also know that this process is longer than it should normally be, and then is not able to separate into two nuclei. Instead, we get multipolar spindles, And I'm not going to show all that data, but instead we're getting multinucleate cells at the end. Okay, you can see here at the end, we can clearly see at least 3 nuclei at the end of the cell division, which is not right. And then those cells go on to senesce and die. So she was seeing this very different thing than what we were seeing. And so we characterized the defect and saw that we were having failure to congress at pro-metaphase. We were seeing multipolar spindles. That's what this guy has. Lagging chromosomes at anaphase and chromosomal bridging, and also the accumulation of micronuclei, which is another sign of a failed mitosis. And so we started looking at our cells as well, and it turns out that while our cells survive, they actually maintain a little bit of this defect. I can't remember which slide I have next. Let's just see. Yeah, well, we'll go in there. So our cells retain some of this defect, even though they're able to survive, and we have a higher than average frequency of cell death, and even those that undergo normal mitosis have a bridging chromosome defect and a long time to complete mitosis. And so that led us to starting to look at what's going on in cell division, not an area that I was an expert in. And so this has been a fun new project for us to work on because we had all these cells that were just trying to divide and they couldn't. And so we figured we had to look at the spindle. And what we saw was, in fact, this is a lot of work that led to discovering methylation was happening at the mid-body as well. So this structure that's in between the 2 nuclei that you can see is a mid-body. And so that's one of the terminal parts of the spindle as you complete mitosis. And we discovered this because we were trying to look very closely at single-cell, high-resolution mitotic events. And the top picture on the left is cells that are stained for H3K36 trimethylation. As I said, that antibody was really good and specific, but it's not totally specific because it also picked up this other mark. And it picked up a mark, which you can see, on the ends of the midbody. So the midbody is a structure that's made of microtubules that are all sort of highly organized and in the end should connect over to the centromeres via the kinetochore and then eventually split out. And here on the right, when we characterize the microtubules for the kinds of marks that they have, we used this antibody for H3K36 trimethylation, and there was a peak on each end. Microtubules are well known to be acetylated, and so with an antibody for acetylated tubulin, you can see that acetylation is more interior of the midbody to the methylation mark, and there's always a dark zone in the midbody, and that dark zone is because there's a large complex of proteins that blocks out any signal. So we're not entirely sure what's going on in the dark zone. But basically we have now methylation happening at the midbody. We spent a lot of time trying to figure out if there were histones hanging out in the midbody. There are not. We ended up making an antibody for microtubule methylation. And it turns out microtubules are a lot like histones. So the histones have a code. Each histone, has these long tails that have all these different marks. We've talked about methylation, but they can phosphorylate and ubiquitinate and a lot of acetylation. So depending on the lysine orientation, the same is true for tubules. And so I didn't know that. And so what we learned, though, is that microtubule, tubulin has also a long tail that sticks out from the bundled microtubule. And this also gets phosphorylated, polyglycolated, detyrosinated or tyrosinated, polyglutamylated. And so this is a schematic that kind of shows that. And it can be acetylated. And what we were able to show using our antibody and some mass spec analysis is that microtubules get methylated on lysine K40. That's shown over here. Actually not over on the tail. But it has a structure that's very similar to lysine 36 on histone H3 in terms of surrounding amino acids. And so we have a switch now on lysine 40 between acetylation and methylation. And the methylation is also trimethylation. And SET-D2 is also doing trimethylation only. We don't know who's doing mono and dimethylation, but it's doing the trimethylation. So back to our cells, kind of proving what this was doing, because now we have two functions. We have SETD2 as a histone methyltransferase and as a microtubule methyltransferase. We had one other thing that we could do to try to separate which of these functions was really important. And that is that we, after we had knocked out in our cell lines, and we've knocked out 4 different cell lines, we then did rescues. So we can rescue our phenotypes with a wild type SETD2. But there are some mutations in SETD2 in different functional domains that are defined by human nature, right? So we look at human tumors and say, where are the mutations? So a lot of the mutations wipe out all of SETD2. They're early truncating or nonsense mutations. But there's a big pileup of mutations in, part of the reason I'm not doing this is because I can't actually see these little things, but I think this is the Sept domain. There's a pile in the Sept domain, which is the catalytically active part of SETD2. That's what does the methyl action, so that makes sense. If you mutate there, methylation dead, you made a dead protein. And then there's a pileup of mutations also in the SRI domain. So the SRI domain is the domain in the SETD2 that's been defined as its interaction with RNA Pol2. So in fact, it's Set RNA Pol2 interaction is the SRI designation. So we made both of those mutations and said, well, let's see what these mutations do. So what you can see on the left is in a panel of mutations, the first cell line is wild type. So it has H3K36 trimethylation. The second panel is the knockout, and so it doesn't. The third panel is the knockout, where we've added back a wild type, brings back H3K36 trimethylation. Fourth panel is the Set domain mutant. It's back to like the knockout. But the SRI domain mutant's been surprising to us. So it surprised us by bringing back H3K36 trimethylation. Behaves like wild type, even in the localization. I'm not going to show you that data, but that mutation, although it does not interact with RNA Pol2, and that's unpublished data, does not seem to have any effect on H3K36 trimethylation. And yet, when we looked at this mitotic defect, and you can see the defect is not as robust as it is in the acute knockout ES cell, or MEFs. But here we're looking at mitotic defects, chromosome bridges and lagging chromosomes. So chromosome bridges are in white boxes. So here, percentage of mitosis. If you get a bridge, you're in a white box. If you have a lagging chromosome, it's a black box. So normally there are not very many. We about double those when we knock out SETD2. We rescue it when we put back in wild type. And we're back to dysfunctional with both the Set domain mutant and the SRI domain mutant. And so it was important to us then to figure out if this SRI domain mutant also has a defect in the microtubule methylation. And in fact, it did. So the bottom is probably the easiest to look at. That's the graph just quantifying the methylation using our methylation-specific antibody, showing when you knock out SETD2, you lose this microtubule methylation. We restore it with the wild type. But both the Set domain mutant, which doesn't have Set domain activity, methyltransferase activity, and the SRI domain mutant both lose methyltransferase. So we're able to separate our functions and show that at least this mitotic defect is unrelated to the histone methyltransferase activity, but is related to the microtubule methyltransferase activity. So there's a lot more work going on in this, but it's all still kind of premature. But our fundamental model is that while SETD2 is a histone methyltransferase, and that's what it's really best known as, when it's absent, the chromatin organization is disrupted. There is a splicing defect that is affected by altered protection of exon boundaries. But there's a secondary activity recognized as microtubule methylation. And both functions can contribute to tumor phenotypes. But in particular, the one that's more likely to be causing tumor genesis is one that disrupts genomic instability. So likely it's the methyltransferase activity on microtubules that's the important part. So I wanted to tell you another story as well, though. So we're going to shift gears entirely. So my lab always has people who work on all kinds of different things. And we have a new project in tumor immunology. And so this came as a result of a new opportunity. It's not just because everybody is doing immunology now. But we moved to Vanderbilt 2 years ago. I only had one postdoc move with me. My husband is an immunologist, and it turns out, although we've been married for 25 years, we really hadn't talked science enough. And so he brought one postdoc too, so we shared a lab for the first year. And in sharing a lab, it turns out kidney cancer immunology is a cool thing to do. So now, with his guidance, we do immunology. So why do immunology in kidney cancer? So there's some obvious reasons. One is that it has the highest CD8 signature of all of the tumors in the Cancer Genome Atlas. These numbers are not so different, but statistically, because these are thousands of tumors, this is really a robust difference. So this is just scoring for CD8 in the TCGA, and it's highest in clear cell. And this is an example when you stain a tumor from clear cell type renal cell carcinoma, how robust the CD8 infiltration is in the tumor, which is quite different than a lot of other tumor types. The other is that this tumor is immune responsive, and now we suddenly have tools to use. And as I mentioned before, I don't have the new data from Saturday's presentation, but it's really exciting what these drugs can do. So the first to be reported was single-agent nivolumab. It's a PD-1 inhibitor, and it improves both primarily response rate, but also overall survival. And again, I've lost my mark on the side, but improves it over compared to everolimus. So again, this is one of those cancer people slides that you're not so impressed by the difference in the curve. But what impresses us as physicians is that, and anybody who's a statistician, I know you're not supposed to make a graph like this, but sometimes patients look like this to you, right? So some patients respond very well and they live for a long time. I mean, this graph goes out 3 years. I have patients easily 3 years on immunotherapy who are doing fine, whose disease has regressed and they're still out there. And so with this sort of surprising feature, so many of us want to understand what it is that's different about some people's tumors from others, and why, if all of these tumors have high CD8 infiltration, why is the PD-1 inhibitor not working more universally? So I like to ask simple questions like I did with chromatin. And so what is going on in there? So to do what is going on in there, we just started a program of harvesting primary tumors from the OR. We get 3 or 5 tumors a week. We process them immediately into single-cell suspensions. We do a little bit of work on the tumor cells themselves, but mostly we're doing this to get the immune cells out. And so we started pulling immune cells and then asking, are they functional? How many are there? What's their distribution? What kinds of T cells, because I'm married to a T cell guy, what kinds of T cells do we have in there? So, like I said, we harvest clear cell renal cell carcinoma and normal kidney. We do single cell suspension and isolate TILs. This data is from a CyTOF analysis of human tumor. This will be of a human tumor. This is a control, showing that in normal control setting, T cells that are resting are normally sort of separated into a bin based on their repertoire of immune features. Up here, this is resting T cell zone. Sorry, no, that's not very satisfying. I need to give it a good name. And when you stimulate, then their features change. And so using CyTOF, this looks at it's like fancy facts. It looks at 70 or so different immune features. So now they fall into a different bin altogether. So the cells really change in phenotype when they activate. Not surprising. So our tumor T cells, though, don't look like either of those. So the T cells that we pull here are intermediate. They actually don't look like they're autophagic T cells. They kind of look like they're halfway senescent based on their features, and they don't respond very well when we try to stimulate them. This just shows sort of the markings. So like I said, they don't respond very well. So they respond a little bit like the sloth from the Zootopia movie. So normally, if you're activating a control CD8 T cell, so it'll go from gray to white. So this is looking at CD25, or you can do CD71, or you can look at cell size. Basically, they respond very quickly. So T cells are a beautiful model to look at activation, and they respond within hours. But when we do this with our CD8 TILs, they're sluggish. They kind of respond, not very much. They don't get nearly the peak intensity, they're kind of So that's why I put the sloth up there. So they also don't proliferate very well. There's a control here on the left showing the proliferation when you stimulate these T cells. So controls will stimulate very consistently. Our CD8 peripheral blood, that's what's in the middle, also is fine. So it's not something that's just genuinely wrong with these patients. It's only when the cells are living in the tumor environment, and then they really don't want to proliferate very much. So we did a lot of work to figure out kind of what's going on. This was sort of the most visually interesting, which is that they have some defects in the way they uptake glucose. They also have defects in their TCA cycle, but their mitochondria are also defective. So this is a picture of mitochondria stained in green. You can also see it here in gray. So the green is mitochondria in a control CD8 cell. And mitochondria normally are sort of co-localized. They're thick, nice, mature. They interact. And so you have sort of this nice, full mitochondria. But in all of our, and this is two different examples, in all of our TILs, they're fragmented. They're really small. They're all over the place, highly dispersed. The total mass is not so different, but the organization of the mitochondria is quite different. And this is just a movie to show, that we're not just taking a static picture. As we move through the cell, you can see the nice organization on the left of the mitochondria of a normal T cell, and then the very fragmented, dispersed mitochondria of a renal cell TIL. We can also see this by EM. So when we look at a control TIL, mitochondria are nice and elongated. They have nice dense cristae. They have sort of all of the good healthy features of mitochondria. And in our TILs, the mitochondria are tiny. They have actually more diffuse cristae. And generally, you see an increase in these small round as compared to nice large rods. So just another validation. And they're dysfunctional, too. So this is measuring the polarization of the mitochondria in these cells in the control. You can see that there's a distribution. So it's red is lower polarization to green is intermediate and blue is high. Most are sort of intermediate in terms of polarization. Ours in the CD8 TILs from the kidney tumors are hyperpolarized. So they're much more in a high polarization range. They also express high levels of mitochondrial ROS and low levels of cytoplasmic ROS. And so what we think is happening is the TIL are making a lot of ROS, but they're not neutralizing it well. And that's in spite of having increased levels of reduced glutathione, which I didn't show here. So they're not managing their ROS very well with their impaired mitochondria. So the question is, can you rescue that? So I think there are two schools of thought that are emerging. One is that we wake up those cells that are in the tumor. Some people suggest that actually they're done and we need to just focus on bringing in fresh troops. So, but we're still trying to wake up the sloth. So the question is, can we reactivate or restore some of the activity of TILs with an SOD2 mimetic? So using a mimetic for the defect in mitochondrial ROS called mitoQ, you can see that we can improve the responsiveness of these tumors, or of these TILs. So this is in response to CD3 stimulation. They're a little bit more stimulatable with mitoQ treatment. Because we think the defect is really primarily mitochondrial, we also can bypass some of the feeding into the mitochondria by giving the cells pyruvate. So we're not worrying about if they can get glucose in or glutamine. We'll just give them pyruvate and let them run the TCA cycle and fuel the mitochondria. And that does it as well. So the defect is somewhere outside of the sort of central functioning of the mitochondria. So they actually respond quite nicely when you stimulate them with pyruvate. So there is an ability to overcome a blockade. What do I have here? Oh, no, that's more evidence that pyruvate can overcome. And so that gives us then a model that suggests that T cell metabolism is disrupted in these tumors. I didn't show you that although glucose is available, they have GLUT1, they have all the things they need to take it up, they don't take it in. but TILs, and particularly the CD8 TILs, just failed uptake or utilize glucose. We're still trying to understand why. The mitochondria are abundant but are small and fragmented, and they're hyperpolarized and produce large quantities of ROS, and that we can partially restore this with pyruvate or ROS scavengers, and that the metabolic defects are contributing to the poor T cell function in the tumor. So ongoing work is looking at understanding this defect better and also understanding the unique contribution of the genetic features of clear cell type renal cell carcinoma that may be contributing to an environment that makes these TILs the way they are. We don't know how much this feature extends to TILs that are resident in other tumor types. We think we're a little bit special because clear cell renal cell carcinoma, as I said at the beginning, VHL mutation, HIF upregulation, defective metabolism in the tumor cells themselves may be making a hostile environment. I will tell you that our initial hypothesis going in was that it was an environment that was sucking up all the glucose, and so these cells were being malnourished or deprived of glucose, but they're not. The glucose is present. It's present in the interstitium, and they don't use it. They just don't know how to use it. So we don't know what the signal is that told them that they shouldn't eat glucose. So how I think about kidney cancer today, I still am a firm believer in VHL and HIF hypoxia biology as the core for clear cell renal cell carcinoma. So everything we do always somehow comes back to that. But chromatin modifiers contribute to an open chromatin and air-prone splicing phenotype. But that SET2 contributes as well to mitotic defects via microtubule loss of methylation. And then in the immune world, the tumor-resident T cells are senescent and suffering from a severe form of malnutrition, but not because they're deprived of nutrients. And bypassing the metabolic defects can restore partial functionality. Is that valuable for immunotherapy? That's on the horizon. So there's plenty to do in the world of renal cell carcinoma, so we love working there. And have just a few, I had these in a different order, but that's all right. I have to thank my lab. So this is my lab today. It's, with my husband. The major people working on this project are Aggie DeCubis in the striped shirt. He did the chromatin biology work in follow-up to what we had done at Chapel Hill. Frank in the front is our microtubule expert. And Peter in the back with the Vanderbilt sweatshirt is the tumor immunologist who really drove that project. Because I knew there would be a lot of trainees in the lab, I thought, and this is kind of left over from my AACR workshop, I think sometimes there's a perception that we do all the science and it's just cool, but kind of in a weird, nerdy, boring way, and we don't have fun. And so we couldn't do this without having a lot of collaborators. And so I happen to have this slide pulled together showing the collaborators that we work with on these various projects. And so I have a very fun set of collaborators. Ian Davis in the blue shirt is the one who got me involved in chromatin, along with Jason Lieb, who discovered and pioneered FAIR-seq. The SET2 microtubule story is all with Cheryl Walker here in the white hair, also teaching me how to play poker, along with Paco, who's a mitosis expert who really helped us know what a midbody was and how you would study it. Our immunology projects are, of course, with Jeff in the middle, Eric Yonash on the side. There's a lot of wine in these pictures. And also samples that we get from colleagues at MD Anderson, Jose Karam and Chris Wood, and also a project drugging some of this that we do with Amado Giacchia, and it didn't talk about. So that's who we do our work with and what we do. So I'd be happy to take questions.
Great talk. Regarding the CD8 TIL work, if I gathered correctly, it sounds like most of those specimens came from nephrectomies, correct?
Yeah.
Do we know if there's any difference in terms of the TIL behavior in primary tumor site versus metastasis?
Yeah, you can ask that question for everything. Because, as a translational biologist, we have so much more access to nephrectomy that almost everything gets done there. We opened our protocol to be available for metastasis specimens, but I think we've analyzed one, so it's way too early to tell. But I think for the genomics, we don't understand the metastasis very well. I think for the immunes, we don't understand metastasis well. Yeah, and then of course that's what kills people, is metastasis, not primary tumor. Very fair. Yeah.
So you showed a lot of the physiology of the cells that would lead me to believe that they would not have a competitive advantage. There's a lot of things that make me think that that seems like that would make the cells die out of apoptosis and so forth. But yet the phenotype is that they are more aggressive, or at least in certain conditions. So how do we
put that?
that paradox?
Yeah, we spent a lot of time worrying about that. So the way I rationalize, so this is completely rationalization because I don't know, is I think that the genomic instability leads to secondary events that give them the competitive advantage that they have. It's surprising to me how consistent it is across different cell lines, so that doesn't totally make sense to me, but I'm not, I know that we're disrupting these cells in a way that's permanent. So I think it could be from the genomic instability and changing your chromosomes and doing duplications and probably more likely deletions. I think that the chromatin could also be an important piece of that too. I actually think that might be more important, and I think that because we can show that with adding back a wild type, we can actually sort of restore that genomic instability phenotype, which surprises me a little bit, but we can. I'm not sure we can correct all of the damage that's been done, but it's hard to put chromatin back. So when you disrupt regional chromatin organization, I'm not sure that gets put back the same way again. So I think that we do some irreversible harm, and that there's probably a selective advantage for some features that let you grow. And I think that's why our cells lived, because we selected for clones, and we were satisfied with a low rate of return, whereas Cheryl's were just consistently dying, and she was seeing the death in real time and really not even trying to propagate her cells. So, yeah. That's what I think. We'll see. Yeah. Oh, we do. Yeah, so we haven't looked at that in as great a detail, but there's a postdoc in the lab who's doing that more now. But they have a similar kind of defect. So CyTOF is really the only thing that we've done there. We haven't done the functional studies with the CD4s. But they have sort of the same features of not activatable very well and kind of having a different phenotype than either resting or stimulated T cells. Yeah.
Is there a significant myeloid?
There is, and we haven't looked at that at all. I have one postdoc who wants to look, but we haven't started that a bit. Yeah, I think there's myeloid. I think based on bioinformatic work, there's some interesting B cell biology as well in kidney tumors. We haven't looked at that at all, but we have a collaborator who's just looking at transcript-based analysis of whole tumors and sees a B cell component. So. All right. Thanks.
Thank you so much.